near the subduction there that you're gonna cover very quickly.
Okay.
Because there's pretty thick sediments on the portion of the plate going down there.
Yeah.
I'm not sure up along the Aleutians. I'm not sure if we have any data up there.
Oh, now I want. I love when that statement gets made. I don't know if we have any data there. I'm like, let's go. Let's have a mission.
I prefer the tropics.
Yeah, I agree. Nautilus gets butt kicked up there.
Yeah.
Big stormy seas are not so much our favorite. So how would you look for those?
Oh, just seafloor mapping.
Okay.
They show up really well as high backscatter in
Right
the side scan, and they're big enough that you can see them. And my preferred way is to have for the backscatter to be black for high intensity, but my colleagues here on the ship don't like that. So
Erin is here shaking her head. Are you on, Erin? Tell us why you do not agree. Oh, you're not on. Yeah, it's obvious why not, she says.
It's an age thing, because all the old guard like me, when we did the initial mapping the seafloor, we're mapping relatively young crust, and relatively young crust is basalt and it's black, and it has high reflectivity. And if it's covered with sediment, like you see down here, it's light colored.
So you wanted, you're visual people.
Exactly.
You wanted to but on a scale from high reflectivity to low reflectivity, your bright white high value is white. Which Erin says, obviously, she's not on a micro now. There's a rumble that's going to go down here in the background. She's like, say high reflectivity black one more time. Say. Oh, man.
I only said that just to
poke.
Sure.
Just to poke.
I've already conceded. But it's my passive aggressive nature just to put it out there.
Awesome. So the ROVs are rising back up to the surface. Just for the little situation update again for everybody, we're ascending back to the ship. We're bringing the biological samples, our rock samples, back up on board. We will plan to dive again. It'll probably be, we're on track still for late tonight. We've about 8 hours of work to do between a transit over to the site and some mapping. That sound about right? 8 hours or so? Midnight. Great. So planning to be back in the water midnight local time, which is coming up about 12 hours from now. We are on Hawaii Standard Time in that time zone here. So site clocks, come back and dive with us then. The ROVs are rising right now at about 17 meters a minute. It's a combination of the winch pulling in the 6/8 cable, which is lifting Argus through the water column, and then Hercules thrusting upwards to keep up so they stay in parallel with one another, or in the same plane as one another coming up through the water. They're turned away, sort of back to back, with the tether stretched out in between the two of them. That keeps the tether from wrapping around either vehicle and gives the backward-facing cameras, the butt cameras, a nice view, making sure that nothing is caught up there. Miles, a question for you. Someone wants to know, is the ship anchored while we are ascending and descending? What's the ship doing right now?
We are in what's called DP. It's dynamic positioning. You could think of it as an anchor. It's tethered to a GPS location, but it is not
anchored to the sky.
Yeah, but not tethered to the seafloor.
Yeah. Sky anchor. I like. Nautilus really doesn't anchor. I'm trying to think of.
I've been at anchor a couple times.
Yeah, I'm trying to think. I mean, it's really just when we're like, we can be sent to anchorage in port, depending on the size of the port and how many berths are available and what the timing of those are. So, but as far as, if you're familiar with smaller boats or dive boats or, your own personal boat, that anchoring is just the standard way to sort of stop and stay in position. That isn't how our ship really works. Yeah, I've spent a couple days at anchor in San Francisco.
In San Diego and outside Galveston.
Oh yeah, Galveston.
Galveston.
We remember that. Now I really want lasagna and to play with lasagna noodles and make crust flexure.
I want graham crackers and icing.
Yes, also great. There's a pretty cool demonstration in those education resources. The hot cocoa earth, if you get like a baking dish or any kind of flat dish, you put milk in it and you put it over a, just like a heat, a heating element. And then you sprinkle, cinnamon or hot cocoa across the top and make an even layer across the top. Is then when you heat and the convection rises and starts to circulate the milk within the dish, it starts to the surface tension of the cocoa or of the cinnamon sort of binds together, and then around those, as the convection currents rise up, it actually breaks the cocoa off it kind of into plates, and the plates start to drift and ram into each other in the dish, which is pretty cool. So you can start to see, essentially effectively a spreading center and then collisions between plates throughout the dish. And then if you stop and, move the whole tray over and apply the heat in a different area, you can have the convection start to rise there and start to push the plates in a different direction.
Cool.
No, but I had meant to ask you the other day when we were talking about, 70 million years ago maybe, as the ballpark, very rough 30 to 80 million year ballpark range on the Kingman seamount. Where were the continents at that point? Is that, some folks had asked, like, is that Pangea at that point, or, what were the dry, dusty parts of Earth doing at that point?
I'm not really sure.
Okay.
I mean, the Pacific was, a little younger. And so the South America and North America were closer to everything. I actually can do the reconstruction to make those available. And because there are some reconstructions online that you can look at that show the location of the plates. That Chris Cotees down at University of Dallas has done a lot of work on those. But there's a lot of them online you can look at.
Awesome.
But the Pacific plate was much smaller, and so was the Nazca, and actually the Nazca in Cocos weren't separate plates at that time. They were another whole plate system.
Interesting. And where can you find those plate bits now?
You could probably just do a now. I mean,
yes.
They're still there. There's North America and South America, and in Australia they're still dry. But they were all just kind of closer together way back when.
Awesome. Are there any crew on the ship that are not scientists? Absolutely. Lots. So our 17 professional crew are professional mariners that keep the water on the outside and the people on the inside and keep us fed and cared for and drive the ship, are all maritime professionals and engineers of some type, chefs, stewardess. It's interesting because lots of our team members have had different types of training, so they've cross-trained, but, Wayne has a physics degree, so that's science, but now is working in a video technology role. Our data engineers are not scientists. Our educators are not generally scientists. ROV pilots aren't often scientists. Our navigators are of variable backgrounds, so much more often in engineering or technology. So many different paths here. And then even within science, I think there's a lot of times when we speak to kids, when we host live interactions, the assumption are that everybody's a marine biologist. And there have been many a cruise we've ended up out here without a single biologist. And we all get along as best as we can, but there are so many different fields of science, branches there to pursue. So the next time you think marine biologist, think marine geophysicist. And then you can ask Rob what that means. I want to see that like written in crayon. I think that'd be the cutest thing in the world. You know, kids like, you ask them when they're little and ask them what they want to be, and they're like firemen or, whatever. I want to just see in crayon scrawl like marine geophysicist. I'm gonna call my nephews.
Said
They'll
no,
write it for
said
me.
no child ever.
Yeah. Oh, I can't hear you. Yeah, what outfit would they wear in the little drawing? I mean,
Lab coat.
yeah.
Passing through
Or like a
250
rock
meters.
hammer, a giant hammer and a big saw. You'd be like part construction worker, part geophysicist.
I think it's like shorts and a polo.
Sandals. Yeah.
A rock hammer and a saw. Same thing an escaped convict would need.
Yeah. Rock hammer and some flip-flops, because safety first. How does the science team get picked for an expedition? And you want to talk about that, Rob? How did you get involved?
I wasn't there to vote.
Last to step back.
Yeah.
Oh.
No, it was actually again kind of like how Miles got his job here. It was just kind of timing, because I was talking to Nicole about something else. Nicole Renault.
Chief scientist for
yeah.
Ocean Exploration Trust.
And she said, "Oh, Rob, we have some cruises. If let me know if you're interested." And it just so happened one was in the Line Islands that I've done some work, and I've also the ports of Hawaii and up here are two of my favorite. So the decision was like 50-50. So I applied, and I guess no one else applied, so that's how they selected me.
Yeah, our science teams apply. You bring up a great point, and we're glad to have you. We're glad you applied. Nautilus's basic exploration cruises, like this one that we're on right now, we have a couple modes of operation. Sometimes we're working with a very specific group to do a very specific goal. That would be the Gorda Ridge or subsea example of an expedition where a team selects and brings their own team aboard and says, this is what we want to do and this is where we want to go. So that's applied exploration, and these are the questions we want to answer. But our basic exploration cruises are sort of the signature and one of the special things our organization gets to do, in that we could get to go to places that have very little basic data, and we want to collect a interdisciplinary team to do it. And those ideas for where we could go do that can come from scientific workshops where we put out just a very basic call. If you had a ship like Nautilus with capabilities like ours, and where would you want to go and why? And they're 1 to 2 page papers. They're very short, and they're available on the website if you want to check those out. OET.org has the results from the workshop, and we just collect a lot of ideas, and that helps us decide where we're going to go. Then when we decide within sort of a year what the schedule will have, what the budget will allow for us to go and do, that's when we start to look for scientists. And the first place we go is people who put an idea out there, who said they'd be interested in a particular area, and then it's about networking. So who do you know that would be a good addition to this expedition? But yeah, we also just there's a lot of volunteering. In September, we'll open up our applications for our ATSI programs. That's another way a lot of people get to join Nautilus, through the internship program like Jake, or our mapping interns, our ROV engineering, video engineering and filmmaking, navigation, ocean science, or our educator program. So no one has ever had like the Nautilus team come, just bang on their front door and be like, "Hi, would you like to go on the adventure of a lifetime?" I think everybody's put themselves out there in one way or another. And if you're interested, apply. Always apply, because we won't know you're interested unless you reach out.
Yeah, Megan, there's actually on YouTube, for people who want to see the reconstruction, there's Earthbite put something out there, and they're kind of like the leading edge of plate tectonic reconstructions.
Earthplate?
Earthbite.
Earthbite. Okay.
BYTE.
Okay.
And the page I'm looking at is plate tectonic reconstruction since Pangea breakup.
Okay.
So Kind of nice, shows you position of all the continents, where the plate boundaries are, and in what direction the plates are moving.
Fantastic. Thanks for that. Earth bite. So as we get up here into the top 200 meters, I will slow down on the questions a little and just let you listen into our operational team over SPL, talking through their workflow to get the ROVs safely back up on deck. So we renovated the wet lab during the last year, so a question came in of how do we decide what branch of science gets to inspect the samples first. The biological samples are a little more time sensitive because they need to be fixed in ethanol, preserving their DNA structure for later analysis without degrading it. But there's a lot more space in the wet lab now, so there's several different groups can all work together and all be in there at once.
Do you have to go to the bathroom? There.
Welcome, Dan. So we're at the watch change, and we're also right at the recovery. So you might hear some other voices come and join. Some people trade in, trade out. Some people run from the control van. I'm really excited we saw a couple of those swimming sea cucumbers. Pelagotheria. I've got one in the slurp jars, but getting to see a couple more was pretty cool.
So am I slowing down around like 50 meters? Is that what we do?
Yeah, slow down for the final approach to 50, and then all stop at 50. That's when it gets passed over.
Okay.
We can go full bore until then. It's all good.
So question of how fast we're ascending. We're ascending about 17 meters a minute, which in a little more than half a mile an hour. So you're walking a lot faster than this. Crawl a lot faster than this. I always think about like 15 meters a minute would be, you know, take one big step, take a 3-foot step, like a lunge-sized step, and then wait for 4 seconds. So like lunge, 2, 3, 4, lunge, 2, 3, and you're like right in the ballpark of how quickly we're ascending. A lunge might even be too big. Depends how tall you are. You only need to cover a meter. Suna, are you back there, or did Alicia
Sure,
tag in?
It's Alicia.
Okay.
Hey, Alicia.
Hi, how's it going?
Very well, thank you.
Glad to hear it.
Can you answer a question for us, Alicia? Viewers are curious a little more about the cataloging and preserving of the samples. You've been through this once, you've gotten to see your first dive sample triage. Talk us through it.
So when when Herc comes on board, we wait for them to rinse it and give us the okay to go take the samples. And then what we do is we take containers and take some of the water that the samples were in, and biological samples usually have the priority because they have DNA and things that we want to preserve. So usually those are the first that we scoop out. Make sure we get all the associates and little critters that were on the, let's say, corals when we pick them up. Then we take them to the wet lab and put them in the fridge. And then we have to photograph all of the samples, biological, rocks, everything. And then we just preserve them in different ways. So some of the rocks will dry out. I don't think we'll be cutting open any rocks on board. But some of our biosamples we will either preserve in ethanol, which is just kind of keeps them as they are, or we'll sometimes dry pieces. Or we'll, take subsamples and cut off little pieces and do different things with them. And then, I think we did fire Niskin. So then we'll empty those out into containers that can be used to look at the environmental DNA in that area, like in that big dense bamboo community that we had.
Did you do any of the eDNA filtering from the first dive yet?
I did not. I think Suna and Jamie did.
Gotcha.
But we'll be doing more today too.
Nice. So queuing up for the recovery here as you're watching, we still have the Hercules camera on channel 1, Argus on channel 2, and if you're watching on the quad, you actually have a view of the control room here. So you're looking at the back of the team, but we'll pause with the questions and let you listen into the ops team.
Tilts all secure.
Great, thank you. And now you're probably good for the rest of it. Sorry, just good to stand by. Don't turn anything else off.
Sub bottom, I turned off.
That's fine.
That's fine.
That's fine.
I'm not using it.
So now you can see the marks in the center there and the two people on deck. They got the tugger lines with the hooks on them, ready to hook. So I'm going to try to line Herc up so that it comes up within the A-frame. Call that getting the goal. I think I'll be able to do it. There's a little bit of current, but I think I'll be okay.
You still getting pushed south?
Yeah, but not unmanageably so. Just slightly.
Okay. Another 15, 20 degrees heading change, we're going to be in the trough.
Yeah, no, this is great. I can't believe I'm still getting USBL hits. And they seem reliable. That's weird.
Wait for it.
Yeah, it'll go, but it's kind of cool.
Also, like, no bow thruster. That helps.
Oh, okay. What's the ship's heading?
255.
Thank you. So once the vehicle's out of the water, Mark will radio up saying you guys are clear to stop your high voltage.
Okay.
You're going to stand by on that until I've secured bypass. And once I've done that, I'll give you the all clear and you can flick the switch down.
Yep.
Could you do bubble on the res, please? It'll sort itself out, I think. Or you might need an iris.
Channel 2, one of my favorite views.
Yeah, I might need some iris action there. Okay, great. Let's just leave it there. Keep an eye on that. Thank you. Why is my, oh, never mind.
Nailed it.
Great view of the jet pump moving around. Fergus view.
Cool. Goal. Yay.
Nice.
So now I'm watching wire cam, camera 11. Keep Argus off the transom. If I pull too hard, it'll swing back. If I don't pull enough, it'll swing in. So.
Yeah.
Hey, Emily, could you tip down on aft cam, please? so we can see all the people on deck and the tuggers and stuff. Thanks. Yeah.
Am I good to turn off the rest of Argus?
Sure, you can do that, yeah. Stand by on the mezzo.
I thought Josh wrote that into the SOPs, but maybe he didn't.
Cool, haven't seen it. I believe you though. Okay, you should be good now. And you can leave GFM and GF check on all the time.
Oh, okay. Yeah.
Yeah, I've yet to see a time when those should be off. In theory it could happen, but I haven't seen it ever. So things I'm doing when I'm getting ready for recovery, I'm making sure my camera's retracted, making sure the arms are stowed, all the boxes are in, and if there's anything on the porch that maybe Mongo's over top of it, making sure it doesn't fly away. That's most of my last minute checks. Making sure I keep the right heading right now, which I'm not.
Couple big rollers here.
So Rennie's down there pulling in the daisy chain, which is taking off that Jacob's ladder knot, separating the recovery line from the tether, and Mark and I think that's Jess have the recovery line on the other side. They're going to tie that to the crane, and then Allison will start hauling in on the crane. You can see a daisy chain on camera 3. Oh, just got pulled out. I timed it well, I guess. So now the daisy chain's in and the recovery line is tied to the crane, I'm going to start moving to my starboard of the ship's port and being in line with this rail as much as possible, maybe slightly outboard of it. The crane will start hauling me in. We'll finish the dive.
Oh yeah. 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 Yeah.
Hi to Corps of Exploration member Derek Hay. Thanks for writing in. Great to have you following along with us. Not here on the ship. I'm glad you enjoyed the dive. Hope you'll join us again at midnight tonight. So our next dive will be midnight Hawaii Standard Time in just about 12 hours.
Right now?
Vehicle power off.
Testing.